Hi all, we are looking at GATE Biotechnology 2022 paper, question number 38. So the question is, match the cell types in column 1 with its function in column 2. So the given cell types in column 1 are option B cells, Q neutrophils, R T cells, and S mast cells. And the given functions in column 2 are 1. Humoral immunity, number 2. Cytotoxicity, number 3. Histamine associated allergy, and number 4. Phagocytosis. So the given options A, B, C, D are different combinations of the options in column 1 with column 2. So we're supposed to match the appropriate cell type with its corresponding function. There are two types of immunity. So it's natural or innate immunity and acquired or adaptive immunity. So the innate immunity is provided by anatomic barriers like mucous membranes, high body temperature, and low pH in the inner lining of our bodies. So blood cells like neutrophils, macrophages, and natural killer cells ingest and destroy the microbes by endocytosis or phagocytosis. And it could also be by the leakage of vascular fluid containing serum protein with antibacterial activity, which act as inflammatory barriers. So, on the other hand, acquired or adaptive immunity is the immunity developed by the host in its body after exposure of antigens, either by naturally occurring infections or artificially upon vaccination, and this is called active immunity. So, passive immunity is acquired after transfer of antibodies or lymphocytes by an immune donor artificially or naturally, as in how antibodies are transferred from the mother's milk to the babies. There are several types of immune cells in our body, each playing different roles at several stages of the immune pathways or reactions. So, there are two major categories based on what precursor cells the immune cells arise from. So the cells arising in the myeloid lineage are thrombocytes, erythrocytes, monocytes, and macrophages. And the lymphoid lineage cells are B lymphocytes, T lymphocytes, and natural killer cells. So let us look at a few of them and their functions in the next few minutes to answer the question. B cells or B lymphocytes, that derive its name from the bursa fabricius of birds, is a key player of the adaptive immune system that is responsible for humoral immunity in mammals. So, B cell production in humans is a lifelong process that starts in the fetal liver intrauterine and in the bone marrow after birth. So, the mature B cells can have 1 to 1.5 into 10 power 5 immunoglobulin receptors for the antigen. So, the B cells also interact with T helper cells and then they proliferate and switch the isotype, that is, they switch the class of immunoglobulin that is produced while retaining the same antigens, and this determines the binding specificity to the antigen. So, the B cells develop from hematopoietic stem cells and they attack invaders that are outside the cells, that is, they attack any foreign antigens. See, on the other hand, T cells or T lymphocytes derive their name from their site of maturation, that is the thymus. So, like the B lymphocytes, T cells have membrane receptors for antigen. T lymphocytes are involved in the regulation of immune response and cell mediated immunity, that is cytotoxicity. It also helps the B cell to produce antibody. So T lymphocytes are generally classified into three types. So they are the T helper cells, that is the CD4 molecules, the cytotoxic T lymphocytes, the CD8 molecules, and the natural killer T cells, that is the NKTs. Class 1 and Class 2 major histocompatibility complex MHC molecules are integral components of the immune system, but they serve different functions and are found on different types of immune cells. So the class 1 MHC molecules are found in all nucleated cells and are recognized by the CD8 plus, that is the cytotoxic T lymphocytes, which get rid of the infected or abnormal cells. On the other hand, class 2 MHCs are found on antigen presenting cells like dendritic cells, macrophages, B cells, and some specialized epithelial cells. So they are present in exogenous antigens, which are usually peptides derived from proteins taken up from outside the cell. So these antigens can be from ingested bacteria, viruses, or other extracellular pathogens. So the class 2 MHC molecules interact with helper T cells, that is the CD4 positive T cells, which play a role in regulating and coordinating the immune response. So then the natural killer T cells, that is the NKTs, are a heterogeneous group of T cells that share properties of both T cells and natural killer cells. So they recognize foreign lipids and glycolipids, and they constitute just 0.2% of all the peripheral blood T cells. These these B and T lymphocytes are involved in humoral and cell-mediated immunity, respectively. Now let's just have an overview of these two immune responses. So, humoral immunity is primarily mediated by B cells and the antibodies produced by the B cells. So, the B cells produce antibodies, also known as immunoglobulins, that circulate in the bloodstream and body fluids. So, these antibodies help neutralize pathogens and toxins in various ways, such as by binding to them and promoting their phagocytosis by other immune cells or activating the complement system. So, humoral immunity is particularly effective against extracellular pathogens like bacteria, viruses in the bloodstream, and toxins released by microorganisms. So, the B cells can also develop into memory B cells, which allow the immune system to respond more rapidly and effectively upon subsequent exposure to the same pathogen. So, on the other hand, we have cell mediated immunity where T cells are the primary components. So, T cells directly interact with infected cells or cancer cells and other abnormal cells. So, they can also help activate other immune cells like the B cells. The cytotoxic T cells, that is the CD8 positive cells, are capable of destroying infected cells or abnormal cells, while the helper T cells, the CD4 positive cells, play a key role in regulating the immune response. So, cell mediated immunity is effective against intracellular pathogens like viruses, some bacteria, fungi, and some types of cancer cells. It's also involved in the rejection of transplanted organs and tissues. So, T cells can also develop into memory T cells, which contribute to a faster and more robust immune response upon re encounter with the same pathogen. The other group of cells forming part of the lymphoid lineage are the natural killer or the NK cells. So, the NK cells are large granular lymphocytes that kill the tumor cells and virus infected targets. So, these cells constitute 5 to 10% of the lymphocytes in the human peripheral blood. So, they do not have membrane receptors for antigen like the T cell and the B cells, but they have NK cell receptors. So these NK cells adhere to infected target cells and induce their cell death through delivery of apoptotic signals by programmed cell death in multicellular organisms. And this is mediated by perforins, which are pore forming cytolytic proteins, and by granzymes like serine proteases released by cytoplasmic granules within the cytotoxic T cells, and tumor necrosis factors, that is cell signaling proteins involved in systemic inflammation, and these are types of cytokines. So fast ligand is a type 2 transmembrane protein expressed on cytotoxic T lymphocytes and natural killer cells. So it binds with the fast receptor on target cells, which induces programmed cell death in the FAS-R carrying target cell. And these NK cells can also kill through antibody dependent cell mediated cytotoxicity mechanisms via cells surface receptors for constant domains present on immunoglobulins. Then there are neutrophils. So, neutrophils are the most highly abundant myeloid cell types, and they comprise about 40 to 70% of all the white blood cells. So, the neutrophils have multi-lobe nucleus and a granulated cytoplasm that stains with both acidic and basic dyes. So, neutrophils are produced by hematopoiesis in the bone marrow, and they are released into the peripheral blood and circulate for about 7 to 10 hours before migrating into the tissues, where they have a lifespan of only a few days. So neutrophils are mobile and they are phagocytic in nature. So, neutrophils ingest, kill, and digest microbial pathogens, and these functions depend on the special proteins found in the primary and secondary granules. So, these proteins are generally enzymes like digestive enzymes and iron chelators and cationic defensins, all of these which help digest the cells. So, the neutrophilic granules contain multiple antimicrobial agents, including oxygen independent lysozyme and hydrogen peroxide and lactoferrin. So these are the enzymes that I just mentioned in the previous point. And the neutrophils dying at the site of infection contribute to the formation of whitish exudate called the pus that you generally get when you get a wound and it's healing. And then basophils are also like WBCs, and they comprise just less than 1% of the total white blood cells. So basophil has a single lobed nucleus and a heavily granulated cytoplasm that stains with a basic dye methylene blue. So basophils are non-phagocytic in nature, but they function by releasing the pharmacologically active substances from their cytoplasmic granules, which play a major role in allergic responses. And then there are mast cells. Mast cells are found in the bone marrow by hematopoiesis and are released into the blood as undifferentiated cells. They do not differentiate until they leave the blood and enter into the tissues. So, these mast cells can be found in a wide variety of tissues, including the skin, connective tissues of several organs, and mucosal epithelial tissue of the respiratory, genitourinary, and the digestive tracts. So, just like the basophils, these cells have large numbers of cytoplasmic granules that contain histamine and other pharmacologically active substances. So, the mast cells, together with basophils, play an important role in the development of allergic responses. Some of the other cells from the myeloid lineage are monocytes and macrophages. So they form part of the mononuclear phagocyte system, that is the MPS, or reticuloendothelial system, or the macrophage system. So this is a part of the immune system that consists of phagocytic cells located in reticular connective tissues. So the mononuclear phagocytic system consists of the two cells again, which are the monocytes, which circulate in the blood, and the macrophages, which are present in the tissue. So both these monocytes and macrophages are involved in phagocytosis or intracellular killing of microorganisms. Further, there are dendritic cells or the Langerhans cells. So these are antigen presenting cells. So the dendritic cell acquired its name because it is covered with long membrane extensions that resemble the dendrites of nerve cells. So there are four types of dendritic cells. So there are Langerhans cells, interstitial dendritic cells, myeloid cells, and lymphoid dendritic cells. So the dendritic cells also arise from the hematopoietic stem cells via different pathways and in different locations. So the dendritic cells again acquire the antigen by phagocytosis. And another type of dendritic cell, the follicular dendritic cell, they do not arise in the bone marrow and they have a different function from the antigen presenting dendritic cells. Eosinophils are another class of cells that belong to the white blood cells of the myeloid lineage, and they comprise about 1 to 3% of the total WBCs, and they are bilobed with a granulated cytoplasm. So they are mobile and phagocytic in nature as well. So they play a major role in the defense against parasites by damaging the membrane of the parasite and an antibody mediated cytotoxic hypersensitivity, that is type 2 hypersensitivity. Now, coming back to our question, match the cell type in column 1 with its function in column 2. So, column 1 has B cells. We just saw that B cells are involved in humoral immunity. So, P would match with 1. So, we have option A and B. And then Q would, neutrophils would are involved in phagocytosis and hence would match with 4. And the T cells are involved in cytotoxicity or cell mediated immunity, and so R would match with number 2. And the mast cells are involved in histamine associated allergies, So S would match with 3. So the answer to this question would be option B. Thank you.